How does the rate of mutation affect the evolutionary process? Can a species' mutation rate be considered fine-tuned for the species for optimal evolution?
Good question. It has a huge implication, and the answer is even better than you're anticipating here. So there you have rates of evolution that are highly variable. HIV eluded our initial ability to do anything about it because it was so rapidly evolving. Likewise, things like cold and flu are so rapidly evolving that, even if we can make some inroads against flu, for example, they're not durable. So in that case, high mutation rate, which has a cost, right? A lot of the mutations that occur, in fact, the expectation would be the vast majority of them make things worse, but to the extent that it discovers some new thing, you can afford a lot of dead viruses if you come up with some new innovation that advances the ball.
Or evades an extant vaccine, for instance.
Right, evades an extant vaccine, or escapes the memory cells that.
Either endogenous or exogenous, our built-in, our onboard immunity, or one that we've received via a shot.
Right. But here's where this gets really interesting, which is even within a creature, the mutation rate is not one thing. And there is clear evidence that different properties are moved around in the genome and altered with respect to their rate of mutation. So you have certain things, like in fact, the adaptive immunity we talked about is contingent on a kind of genetic shuffling that takes place. So basically there's a random number generator-ish phenomenon in the genome. There is also the ability to move things on chromosomes where they are more and less prone to be disrupted and therefore to change in ways that could be selected. So, for example,
like closer to the chiasma, more and less susceptible to crossing over.
Right, exactly.
Okay.
So I learned from Bill Rice, who we mentioned on the program last time, that the fastest evolving gene known at the time, and I don't know that it's changed, the creature may have changed, but the likely the process has not. The fastest evolving gene known was the gene for the acrosomal proteins in, I think it was abalone, if I recall correctly, in the sperm. And the idea is that there's a...
Right, so the acrosomal proteins are, is that tail proteins, or is that like the coating?
No, it's the protein that enters the egg.
Yeah, that's what I thought.
There's a kind of arms race between males. The one that activates the egg first has an advantage, obviously, because it shuts down the egg's availability to other sperm. So where there's sperm competition, there's intense selection for a better formula. Mm-hmm. And it may be that this is part of an arms race where there's actually resistance on the part of the substrate. There's evolutionary resistance on the part of the egg. And so anyway, rapidly evolving proteins in acrosomal proteins in sperm. Slowest evolving genes known are those for histone proteins. Yeah. Histone proteins being proteins around which DNA is wrapped, partially just to keep it organized so it isn't a huge tangle that breaks up and can't function. But we have learned more recently that the way in which proteins are wrapped around the way in which DNA is wrapped around these histone proteins, which look like rods and balls, and they're packed in a very regular form. In fact, you can see it in an electron micrograph. You can see, you see these, I forget what they're called, nucleosomes. But anyway, you see these regular balls that are created by the way that the DNA is wrapped, and then there's sort of a fractal process where it's wrapped, and then it's wrapped at a higher level and higher level. But anyway, the way in which the genes are wrapped around the histone proteins dictates whether or not they are available for transcription. And so what this means is that this is a mechanism for turning genes on and off. So if you think about the fact that your eye has all the proteins necessary to make your liver work, but most of those aren't useful in your eye, so they have to be silent. So you can wrap genes in such a way that they are effectively invisible to the cell and not interfering with anything. They're dormant. So the point is, in histone proteins, these are extremely ancient structures. They go back to the beginning of eukaryotes, which you labeled as how far back year-wise?
1.2 to 2 billion years.
1.2 to 2 billion years. That's a very ancient structure. And the idea is any alteration you would make to a histone protein is almost certain to be catastrophic, and not just catastrophic for some process, but catastrophic across the whole genome, because everything is dependent on being well wrapped and organized. So anyway, very slowly evolving. So
yeah, lowest mutation rate there.
Yep.
Very cool.